Today I'm going to tell you all about some of the work that my lab has been doing for the past several years on the topic of engineering anaerobic consortia that originate from the guts of herbivores for lignocellulose breakdown. And in particular, I'm going to focus on some of the more emerging stories that have come out of this research in the past several months and things that we're working on. This is our beautiful campus, which I miss being at every day. So for those of you who are looking forward to traveling again, come see us in Santa Barbara. Our COVID-19 cases are plummeting, and things are looking up, at least. So something to be grateful for. The other thing I'll mention, and many of you know me well, but you may not know that the pandemic has been very interesting for me. How have I spent the pandemic in addition to all of these challenges that we all face? I had twins right in the middle of all of this. So this was me in front of the Santa Barbara Cottage Hospital on April 13th of 2020. And my husband and I emerged with these two little beauties to join their about to be 4-year-old brother. So as you can imagine, things have been very interesting for us. I also wanted to mention this not only to kind of keep things real, but also to explain any of the noises that will be coming behind me or children entering this room, because it is technically a holiday here in the US. It's Memorial Day, and nobody's in daycare. So on the topic of children, it's a pretty easy transition to talk about this. So poop. It's kind of a big deal. It actually inspires a lot of what we do, not from a gross point of view, but it's actually a pretty good reporter of microbes in organisms, right? There's a whole bunch of companies that are looking at mining in fecal material to think health outcomes. Here's some of the companies that I just kind of grabbed that have started to really put a foot in this area, trying to bank microbes in fecal samples, trying to develop fecal-based therapeutics, et cetera, et cetera. And some of these are old tried and true companies. Some of them are, many of them are emerging startup companies getting into this world. But now more so than ever before, there's a reason to look at the digestive components of organisms, everything for treating human health to, of course, mining for what this community is very interested in as well, new biotechnologies, specifically gut microbes that do really interesting things, right, that do cool chemistry. So from my point of view, we're really interested in gut microbes, specifically from herbivores, and I'll motivate that in a second, because they do the remarkable chemistry that their host organisms cannot accomplish, right? And they do that in a team-wise format, so they are dividing and conquering really difficult chemical tasks and passing chemicals and messages between each other all the time in a dynamic fashion. Just like people, microbes are what I like to call friends, enemies, and frenemies, and really all of the social dynamics in between. It's well known that many microorganisms rely upon each other in order to function, and because they might not have all of the necessary metabolic capabilities to live in a certain environment, so they rely on a friend for some sort of metabolite here and there. Often microbes are competing for the exact same resources and need to keep each other in check, right? And some microbes, of course, will change their dynamics depending on the environment, depending on the substrate and food sources coming in, and really a whole bunch of other factors that we and others are yet discovering. But I'm an engineer, and so I'm really interested in addressing some of these seemingly very basic questions of who is there, right? So who's in the gut microbiome? And with that knowledge, can we say anything about what those microorganisms might be doing on a metabolic basis? And again, in the spirit of trying to engineer these systems, what I want to know is how we can learn from nature, specifically how nature puts these microbial communities together in effort to design microbial communities that can work for us or do a very specific task. Okay, so looking at the problem here, just saying, okay, Just making sure I'm not missing anything. So what is our problem that we're rallying around? So this problem of taking the non-food part of plant material and turning that into something that I'll call value added. This is a long-standing problem that certainly we are not new to to thinking of these types of challenges, and many of you share this inspiration as well. And so if you look at the schematic of plant cell wall material, what you'll see is that you need to have many different components. I'll just click through these because this community knows this exceptionally well. You need a battery of different enzymatic players to break down crystalline cellulose, hemicellulose, and other plant cell wall components, including lignin, which, depending on if you're a glass half full or half empty person, is really an opportunity or a huge impediment. In biotechnology, this is right now a huge impediment because the lignin acts to keep many microbes and their enzymes specifically out, in order to give the plant, sort of a fighting chance, if you will. But especially from a chemical engineering perspective, if I look at lignin, I see a lot of really expensive chemistry. And so if there's a way that we could break lignin down into so-called bite-sized pieces, especially regular fractions of chemicals, then that could be a recipe for chemical upgrading, for making coatings, for drop-in fuels, for maybe even making renewable polymers. But the tried and true message here is that substrates like this are an absolute mess. As many of us know, these lignocellulosic substrates vary in the relative content of these polymers from plant to plant, from growing season to growing season. And this is done somewhat intentionally because nature is very smart at adapting to the changing conditions that a plant has to face. But nature has also given us solutions, right? So there's many different places where you could think of sourcing microbes and these powerful enzymes that you need to degrade lignocellulose. And so we've gotten very interested in the digestive tract of herbivores, which grow very big by grazing on these very same crude sources of lignocellulose. And if you peer inside of what's in the rumen microbiome, and again, there's many studies that have done this, you find, of course, a changing environment, a dynamic digesta that's mildly acidic, that the herbivore is changing its what it's eating, and so the digesta and the microbiome is matching to that challenge. Of course, much of this is anaerobic. I think something that is starting to be appreciated in the past several years is that this is actually a pretty dynamic environment, and that it's not just a home to bacteria. Of course, bacteria overwhelmed by numbers, but if you start to look at some of the other players, you can see, and these are kind of wild estimates here, but there's methanogenic archaea, which obviously play a key role in methane release. There's protists, which I would argue we have the least information about what they're really doing in these types of systems. I'm not going to talk about them today, but sort of unsung heroes of these communities are the anaerobic fungi, which are there as really, by the numbers, very minor players, but they are big. So if you start to actually think about biomass by gram wet weight or dry weight, they start to compete a little bit with bacteria. But by the numbers, they're there as sort of somewhat minor players. You can actually eliminate them completely from the digestive tract of herbivores, and you'll still see that they can eat lignocellulose. So there's something to this competition and this diversity that I would argue is really important, right? And so from our perspective, we wanted to study these systems to develop bio-inspired technologies for breaking down lignocellulose. In particular, can we build microbial communities that preserve this important kind of cross-domain interaction and this diversity that's seen in natural systems? And in doing that, there's kind of two canonical strategies to accomplish this goal of making consortia that you could plug into a bioreactor and start to divide and conquer these difficult steps of biomass breakdown. One that I will primarily talk about today is what's called this top-down approach, where you take, let's say, a natural source of these microorganisms, you apply some sort of selective pressure that might be enriching certain microbes with lignocellulosic substrate so that you're only getting a minimal system of microbes that are tuned to break down lignocellulose. And then you have this minimal system that is connected by mutualism, commensalism. These are just two examples. You could have antagonistic interactions, etc. But nevertheless, you have this minimal system that you can play with in the lab. The downside of achieving a minimal system this way is that generally you have almost no idea what you're working with. You have to do all of the bottom-up characterization, genomics, transcriptomics, etc., to really understand what microbes are underlying the degrading activity, and chances are very, very good that you will everything you're working with is novel. The flip side, and which is really, I would argue, kind of the more engineering route, which it's ironic that we don't really use this, but is assembling some truly synthetic consortia from the bottom up. What that means is that you go to a strain data bank or your favorite microorganism that or chassis strain that it has maybe even a genome scale model associated with it, maybe even genetic tools, and you put those together based on some envisioned complementary metabolic interactions, and you hope that these seeming marriages take, right? The upside here is that you know everything that you're working with. You might even be able to draft a nice computational model to understand how it might work. The downside is that in almost all cases, these synthetic consortia fall apart after a couple of consecutive batch cultures, and then you're kind of left with nothing. So because of that very strong limitation, most of the work that I'll tell you about today is what I would categorize in this top-down microbial enrichment strategy. Again, we're trying to get microbial communities that can degrade lignocellulose. So where do we go to do that? We actually go right to the source. So here's my colleague who is over in the UK, my former student Sean Gilmore, scooping up fresh droppings in the draft pen at the local Santa Barbara Zoo. Lucky for Sean and all of my students, you don't need very much in the way of samples to actually extract anaerobic biomass degrading organisms. Here I'm showing just a smattering of different fecal samples that we've obtained from zoo animals that are eating these lignocellulosic substrates. I'm skipping a lot of steps here, but essentially we use anaerobic technique and roll tube technology and also dilution to extinction to get unique isolates. We can also do this for consortia, so this is the same pipeline. But I thought I'd just take a minute and show you how we get anaerobic fungi out of these systems by suppressing bacterial growth with the use of targeted antibiotics. And these are 4 different strains of anaerobic fungi that our group has discovered and named. The names are not quite as important. I can tell you about how we named them, but we don't establish the genus name, but the species name we've had some fun with. So for example, this Pyromyces finis came from a horse called Huckleberry Finn. So you can see we have fun with names. The names are not as important as actually looking at what these microbes are doing. So if you look at the fungi themselves, hopefully you can see in each of these pictures their morphology is just a little bit different, but these are all growing on crude lignocellulose. So you can see this is a great one of Pyromyces finis, where you see the plant biomass and the fungus really enveloping that biomass and literally using its rhizoid or root-like system to punch through the plant biomass, and that's where we're thinking that enzymes are being secreted. The same is true of this Anaerobes robustus, which has a slightly different morphology, Neocalamastis californiae, and Cecomyces cherobus, which is our newest isolate that we're about to release the genome of. We grow all of these in anaerobic batch culture. So many of you may have seen this already. This is several years old now, but the first thing that we did was to really sequence the transcriptomes of these novel isolates and establish that they actually have a huge equipment list of carbohydrate active enzymes, including components of fungal cellulosomes. And so for those who are not, have not maybe heard me talk before, just a quick step back of what these anaerobic fungi are. They're early branching fungi. So this is the fungal tree of of life, and I'm pointing at the Neocalamastico here. This is from the DOE Joint Genome Institute. I have on this slide that there's 9 characterized genera, but others would argue that there's 20 plus, and I would not argue with them. Clearly, there's a need to characterize more of these genera. I would argue that we should have, as a joke, that we should have fewer people from Wales naming naming these systems, because I can't pronounce stuff like that. So it's a small plea from a lowly American here to please try and use some names that I can pronounce. But that aside, how these reproduce is we have these motile zoospores that are literally swimming with purpose to a place that they like, a surface of lignocellulose, where they're starting to germinate these rhizoid networks that are going literally punching into that plant biomass, all the while secreting enzymes. And if you've never heard about these fungi before, I would chalk that up to the fact that there's just a small but thankfully growing community of researchers that are looking at these. And they have suffered from a lack of genomic characterization or available strains. This again is changing, which is great. And they've been difficult to cryopreserve. They're not yet quite genetically tractable, but we and others have been working on this. And when we started this project, there was really no proteomic characterization of the powerful machinery that underlies the biomass degrading abilities of these fungi. And this is what they look like. This is a helium ion microscopy image showing these anaerobic fungi literally punching into the plant biomass. It's differentially colored, so you can try and see the fungal mat on top of the reed canary grass in this case. So one of the first things we wanted to do was to see what these enzymes were, and specifically to hunt for novel enzymes. One of the key experiments that was done by Kevin Solomon when he was a postdoc in my group, he's now leading his own group at the University of Delaware, is to use transcriptional regulation patterns to discover novel enzymes. Essentially what that means is that we don't think that these fungi are going to make a ton of these key CAZymes and other machinery when they're growing on glucose, right? But that need changes when they're growing on cellulobiose and filter paper, abasil, reed canary grass, lignocellulosic grasses, right? So you can see that if we look at different classes of enzymes and how those are enriched via RNA sequencing, as the substrate becomes more complex, we can see that yes, there's starting to be more of these key groups firing as we are challenging with a more challenging substrate. So in doing that, oh, this is out of place. Okay, I'll go back to that. So one of the things that we wanted to do is to essentially use these regulation patterns to decipher what these novel enzymes. And so what we did is we grew these fungi on reed canary grass to fire all their enzymes that would be associated with biomass breakdown, or at least as best as we could. And then after they've sufficiently done that, we add in glucose and essentially expect that the fungi will rapidly repress the expression of these CAZymes. In doing that, this is exactly what we saw. So everything that goes up in red is associated with metabolic reprogramming or stress response, but all of these individual transcripts in blue are associated with plant biomass breakdown, or at least 80% of them that we knew. And the majority of these downregulated genes are genes that we could annotate as putative CAZymes. And we asked the question, okay, so these other things that kind of fell in line that we don't know what they are there new enzymes or that are either directly or indirectly playing a role in biomass degradation here? And the answer is yes. So for example, we've worked with J-Bay and Andrei Joachimiak at Argonne to find some glycosyl hydrolase 5. This is a crystal structure that's not yet published, but we've also found components of fungal cellulosomes, which is this slide that was vaguely out of place. So you've probably heard of cellulosomes from anaerobic bacterial systems. Short story is that fungi have them too, but the parts are all different, similar in concept only. So these giant megadalton biomass degrading complexes. Actually, I need to update this. Apologies, this has been long published in 2017. It's also in Nature Microbiology if you want to see more information on this. So when we sequenced the genomes of these fungi, we found 400 putative Dockerin, fungal Dockerin-fused genes. The Dockerin domain is key at plugging these enzymatic domains, these CAZymes, into a flexible scaffold. We didn't know in fungi what the scaffold was, but we did find a whole bunch of repeat-rich putative protein scaffolds that we thought could be responsible, that are unique to the anaerobic fungi. So we asked this question of, can we actually test whether or not this putative scaffold and these Dockerin domain proteins are actually sticking to each other? Long story short, we can by making these domains recombinantly in yeast. We find that by ELISA and even by SPR, they are binding to each other, and we can wreck that binding by eliminating key binding motifs. So that was and one of these, actually all of these proteins are contained in this data set as well, which is why I had this out of order. And so with that, I'll take just a humor break and say that we get a lot of attention for this, a lot of media, and I've frequently found myself asking people not to send us poop, although you all are from the community that if you have really interesting samples, particularly that have anaerobic fungi associated with them, I'd love to hear from you. We love to hunt for new microorganisms that do this amazing chemistry. And something I always tell trainees is that anytime a reporter comes and asks you about your work, make sure you proof the story before it goes to print. So I will leave that at that, and transition into what we've actually done with communities. So I've told you a lot about how we've gone into herbivores and isolated anaerobic fungi, and why anaerobic fungi are really interesting to source as biotech platforms to identify enzymes and identify completely novel enzymes. But we were really interested in the communities of these microorganisms and how these teams of microorganisms can do really fantastic things. And so we went back to the zoo and visited a San Clemente Island goat whose name happens to be Elway. And we actually wanted to enrich for these microbial consortia on different flavors, if you will, of plant biomass. We used 4 different targets: alfalfa, bagasse, canary-grass, and xylem. And then in order to get diverse teams, which was really what we're after, we needed to help these communities by adding chemical help in the form of antibiotics. Because if you just enrich on these substrates, all you're going to get is bacteria. So we had one condition where we enrich for a fungal consortia. So we add chloramphenicol to permit only growth of fungi. We had another where we knock down, but not completely eliminate, the action of bacteria, but with a cocktail of penicillin and streptomycin, which should enrich for fungi methanogens, and another one where we just let the microbes fight it out. And we carried out 10 consecutive batch cultures in triplicate on each of these biomass types, and tracked in as close to real time as we could metabolites, so chemical metabolites that are coming off in the liquid and the gaseous samples, fraction of the headspace, and also doing metagenomics, so taking the DNA and ultimately the RNA from each of these samples and looking at how these different taxa are changing with time. And in doing that, we partnered, of course, with the Joint Genome Institute in order to do all of this. But essentially, we tracked both marker gene analysis, which is not nearly as powerful, but the unique contribution of this work, I would say, is really in the metagenome pipeline. And I'll get into this if anybody's interested, but essentially we're putting the genomes together from scratch. And we're doing that specifically to fish for metabolic potential of these communities. In doing so, we were able to put together about 700, a little bit more than 700 prokaryotic metagenomes. 96% are novel species. That's not surprising. About 200,000 carbohydrate active enzymes just in the system. So I realize this is a giant set of data, but essentially what we're doing is we're annotating CAZymes in all of the different spokes, which are genomes. And this is a log scale, but you can see easily that the fungi are a minor population of the system. I should say these are eukaryotic mags, of course, and not prokaryotic mag genomes, which we also were fortunate enough to somewhat assemble, not well. But they do have the lion's share of enzymes by the genome, right? But you see other players, there's Clostridium, there's Rhinococcus, there's a whole bunch of other usual suspects in the system. And of course, you know, not surprisingly, most of these microbes we didn't manage to enrich by cultivation, and these were in the feces. But those that we did started doing some really interesting things. So these 3 chemical treatments that we had to kind of sculpt the membership, we were also looking at the off gas and products that were coming out of these systems. Not surprisingly, a system that only has fungi is not making methane because there's no way to do that metabolically. They're just making hydrogen. But the really interesting difference is seen between these fungal dominated consortia and the bacterial dominated consortia with methanogens present, where you start to see that actually in the fungal dominated system, there's almost twice as much methane that's being generated when the bacteria are not down. We were really intrigued by that, wanted to know what it means. And so we started making food webs to figure out how these microbes are passing, putatively passing along different substrates. So each microbe here is the shape that comes from enrichment on one of these target substrates. And if there's a connection here, that means that it produces CAZymes to digest cellulose into hexose sugars or hemicellulose into pentose-based sugars, etc. You can sort of see how things flow. And not to parse everything about this data, but you see there's actually a wide spread of different chemistry here on the path to methane and some other terminal fermentation products. And this is in the bacterial-dominated consortia. But when you have a fungal-dominated consortia, this is really streamlined. You have fewer members and fewer terminal products. So there's a much more direct path to methane, which is part of why we think that these fungal-dominated consortia are making more methane. And so just to wrap up here, what have we actually done with this? So in learning kind of these members of minimal systems and consortia from herbivores, it's an opportunity to really build consortia that we can now start to play with from a synthetic biology perspective. So one way that we've done that is by pairing anaerobic fungi and methanogenic archaea together based purely on this common metabolic connection point of taking hydrogen gas that's produced from the fungal hydrogenosomes and using that as a source for methanogenic archaea to thrive and make methane. So this is a form of mutualism, and then commensalism, so taking opportunists that just want to eat the sugars and putting them into synthetic culture. So in doing this, and this is from a public paper that we published in UCS SynBio a couple years ago, we were able to take some anaerobic fungi and some Methanobacteria barentii and put them together in synthetic culture. They make methane. Of course, they're not very stable, but it's an opportunity to actually start to look at some of the underlying impacts that these microbes have on each other. And doing that, we can actually look at how they regulate their genes due to the presence of each other there. The surprise there is that the vast majority of genes that are regulated are associated with biomass breakdown in the fungi, even though the methanogens have no direct role in that process. And in particular, the fungi are upregulating cellulosomes when the methanogens are around, which was a surprise to us. The other thing that we can do is to actually make a bit of a bioprocess out of this by taking anaerobic fungi or anaerobic fungi-driven consortia and sweetening up a liquid broth by essentially letting them degrade things into sugar. They produce way more sugar than they need to survive, and then adding in metabolically engineered microbial chassis like yeast and E. coli to convert that sugar to commodity chemicals. So I'm showing this for yeast. Essentially, these fungi are releasing a lot more glucose than they need, and if we drop in yeast, they're actually able to convert that into biomass in this case, but also other fuels, chemicals, and value-added products. So this is one way to use these microbes for a biotech purpose. And just really quickly to wrap up, we also wanted to know what else is hiding in these genomes. So I mentioned that there's a small community of researchers that are working on on these fungi, but it is growing, and there's starting to be a lot more genomes that are available as a general resource. One of the things that we identified rather quickly is that there's actually a huge amount of not only carbohydrate active enzymes, but the transporters that give these fungi the ability to take up certain sugars, and also potentially resist other microbes in their community. There's a lot of receptors that have non-canonical architecture, meaning that they look like they're fusions between prokaryotic substrate binding proteins with eukaryotic domains. Very, very interesting from an evolutionary perspective. If anyone's interested, I'd be happy to talk more. But we've also functionally expressed many of these in yeast. So we've taken sweet transporters, for example. This is a paper that just came out in Metabolic Engineering from the anaerobic fungi, and put them in Saccharomyces cerevisiae, and they help them to actually not only intake more sugar, but specifically uptake more xylose in a wild type strain, which is great. And another paper that just came out in PNAS, where we noticed that quite remarkably, and also very surprisingly, these anaerobic fungi seem to be producing the building blocks of polyketide synthases, non-ribosomal peptide synthetases, bacteriocins, terpenes, etc., building blocks of natural products. And so what we did is we essentially scanned these genomes and looked at how transcriptomically active they are, and found some putative products. So this is the first time that the anaerobic fungi have been mined for natural products. So that could be another potential use for them. So with that, I'll wrap up and say that I hope that I've shown you in this smattering of different stories that anaerobes are very interesting to study, both for CAZyme potential and these other more interesting directions that have come from this work, specifically anaerobic fungi and the communities that are dominated by them. And we are now building natural partnerships and synthetic partnerships from these microbes to drive lignocellulosic bioprocessing and value-added products. And so with that, I'll thank, there's a paper that just came out, and the goat. I'll thank all the folks who actually did the work, U.S. Department of Energy for funding, along with all the rest of the folks here, and happy to take questions if there's time.
Thank you, Michelle, for a very nice talk, very inspiring. Yes, there is question for time, so please sign up on the list. First one out is Vincent.
Hi, Michelle, a very nice talk, thank you. I was just wondering for the anaerobic fungi, two potentially related questions. Could you make a few more comments on how easy it is to culture them? And connected to that, and in a more ecological context, how well and in which manner do these anaerobic fungi deal with oxygen? I mean, how sensitive are they?
Yeah, so the cultivation part, it's one of those things that many of you probably also know. You can read a protocol about it, and you can try and do it, and you'll have kind of some luck, and sometimes you won't. It is immensely easier if you just are next to someone who's doing this, and if you have a good master of anaerobic technique, it seems to be pretty easy. The media formulations for cultivating these anaerobic fungi are not trivial, so you need things like clarified rumen fluid, very redox balanced conditions, redox indicator. Mike, familiar with anaerobic technique, that's not that big of a jump. But we've hosted several people to try and teach them how to extract and enrich these fungi from natural sources, and cryopreserve them, etc. And we're always willing to do that. But yeah, we do have several protocols out there, and we're always willing to help people to do this. To try and grow the community. In regards to the oxygen tolerance, so that's actually been a bit of a surprise. So we have, these are obligate anaerobes, but they seem to be somewhat microaerotolerant. So what that means is that they don't seem to thrive or grow at all when oxygen is around, but they do seem to have some methods for dealing with it, so that they're not nearly as oxygen sensitive as like what methanogens are, for example. Methanogens just know that oxygen is around and they're just done. But the anaerobic fungi, particularly the spores, we can keep them alive, 30 minutes to an hour when in just normal kind of oxygen concentrations, and that has been a bit of a surprise for us.
Thanks.
Any other questions? I was really curious about the, it's really nice work. I'm really impressed. I was really curious about the productions that you're doing right now. What kind of scale are you working on?
Yeah.
For these pilots?
I'm but a lowly academic, so anything that I say, about scale up or anything, I mean, we're working on 1-2 liter scales in our lab. This is where I've been very excited to partner with the Joint Bioenergy Institute, JBay, which is a U.S. Department of Energy Bioenergy Research Center, because they actually have a nice pathway for scale up that's pilot scale. So we have talked about trying to transfer some of this into their bioreactors, but of course, anaerobic organisms and scale up for those, it's just kind of flies in the face of how you would do scale up of virtually any other organism that consumes oxygen. So we're still working on that, but yeah, we have ourselves not done anything past like a couple of liters of scale. So it would be very interesting to translate these organisms into some properly scaled up bioreactor. That said, I mean, people have used enzymes that are sourced from these anaerobic fungi and scaled bioreactors, and they've done quite well because you can express not all of them, but several of them in E. coli and yeast and other kind of industrially friendly chassis organisms.
Do you have another question, Vincent?
If I'm allowed, if there's no other questions, I have one more.
Yes, yes,
Yeah,
please.
I was just wondering, Michelle, about the enzymes. I mean, you introduced your talk with the promise of very much novel enzymes and potentially unseen things. You did perhaps not say so much about that. Could you give us a little taste of?
Yeah.
I mean, what do we know? What is coming out of this, you think, on the enzyme side?
Right. So with the enzymes that we've identified that I would classify as more novel, I mean, so one is that I kind of glossed over this, but there's a huge amount of horizontal gene transfer between anaerobic bacteria and anaerobic fungi. So they have a lot of genes that you just wouldn't expect to be there. So that includes more bacterial looking genes. Actually, a number of the fungal cellulosome genes look like they were horizontally transferred from bacteria that produce cellulosomes. That's a very interesting story. But to get right to the most interesting part of it, lignin active genes. So we have identified a whole bunch of very surprising genes that are in cellulosomes and then also not, that give the fungi the ability to anaerobically depolymerize lignin. And I can say that irrefutably now because we actually do have the evidence, just isn't published yet, which is like the worst thing to say on a conference because I want to show it. But yeah, we have 2D NMR data that has finally shown us that lignin is not only liberated but maybe even catabolized by the anaerobic fungi. And by combining a whole bunch of omic techniques, we have a suite of responsible enzymes that we think are doing this. Are we close to a mechanism? Not at all, but we do at least have that. That is very exciting work that we're working on with JBay right now, that we're hoping to publish in the coming months, because now we finally have the irrefutable evidence to suggest it. But I just wish I knew what the mechanism was. I can tell you what the players are, but I think the mechanism will be a long, long time coming.
It creates a lot of nice work for many of us because, of course, anaerobic lignin conversion is, of course, very, very interesting. Yeah. Thank you.